Do you also wonder how complex mysteries are solved with the help of small clues like blood, hair, or few cells? Yes, in this video I'll show how it's done. Any two person on the earth have their 99.9% of DNA exactly the same. But what about the remaining 0.1%? This 0.1% of DNA may be useful in determining similarities and differences among different people. By using above fact, a famous geneticist Alex Jeffrey discovered the technique of DNA fingerprinting in 1984, which can be used in solving criminal scenes. In this method, we need few cells, hair root or blood samples collected from crime scenes. Now let's look deep into this matter. There are few sections of DNA where instead of a gene coding for something, we have a non-coding part. This non-coding part of DNA consists of sequence of 3, 4 or 5 bases that are repeated many times. For example, ACG, ACG, AGTC, AGTC, GATCA, GATCA. The sequence is called variable number tandem repeats or VNTR in short. Now let's discuss the process. Firstly, DNA is extracted from cell sample found from the crime scene. This could be hair roots, cells or blood. This DNA is then cut into small fragments by enzymes like restriction endonucleases. The sample is then poured into gaps of agarose gel for the process of gel electrophoresis. This separates the DNA fragments by size. This pattern is transferred to nitrocellulose membrane in southern blotting. Probes are applied and then X-ray is passed, which allows us to see the pattern. The pattern is then compared with the suspects. The one having the pattern identical to the pattern found from the criminal scene is found to be guilty. In this way, complex mysteries can be solved with the help of modern day technique of DNA fingerprinting.